Greetings, Professor. We are the team formed by Oliver, Eric, Fatima, Andrea, Martin, and Valeria, and we are going to present our second delivery for the reactor design project. First of all, we need to remember the scope of our problem. Our team is searching to develop a reactor for the ethanol production from a sugar-based feed. The name of our reaction is fermentation reaction.
Starch and sugar-based ethanol production is mostly produced in the United States from starch-based crops by dry or wet mill processing. Nearly 90% of ethanol plants are dry mills due to lower capital costs. Dry milling is a process that grinds corn into flour and ferments it into ethanol, with co-products of distillers, grains, and carbon dioxide. Wet mill plants primarily produce corn sweeteners, along with ethanol and several other co-products, such as corn oil and starch. Wet mill separates starch, protein, and fiber in corn prior to processing these components into products, such as ethanol. This process requires a high conversion and is usually performed in batch reactors. To design this reactor, we need to consider several things like cost of production, offer, demand, and the place where it could be produced. The demand for ethanol in Mexico is very big because per year Mexico demands 39.2 million liters of ethanol. This means that our reactor and our reaction is viable. To operate this reactor, we are going to use several standard batch reactors in parallel and assume the reactor dimensions shown in the presentation. For the calculation of the electricity needed for mixing conditions in a single batch reactor for 6 rectangular vertical pallets and using the optimal criteria for the diameter of the propeller being 1/3 on the total reactor volume and calculating the number of Reynolds for a mixture flow, we obtain that the data needed for the equation to calculate the power and multiplying by the time we obtain the cost.
The next step for our project development is to choose the correct ideal type of reactor for our reaction to be carried out. We chose a batch reactor. The reason why is because we consider this type to be the best since we are searching to obtain the maximum conversion for our process, and that is the perfect usage for batch reactors according to Fogler and Levenspiel. To make the proper calculations, we must establish the conditions for our reactor, which will be a temperature of 30 degrees Celsius, pressure of 1 atm, anaerobic conditions, which means no oxygen. The mix needs to be continuously stirred, and a pH value of 5.5. We must consider for a batch reactor there are no flow rates, heating conditions to maintain the temperature at 30 degrees Celsius.
Now that we know our condition, we can begin our mass balance for the reactor system. First, we establish our consideration. We need to consider the total annual production of ethanol in Mexico, the theoretical conversion of the global reaction, some specific assumption for the operating condition already described above, the special assumption that our plant will be required to produce 10% of the total Mexico production and the initial concentration, which will be explained in a moment. Our input is considering previous experimentation made by Siqueira et and others, where they conclude that the best concentration of total solid in solution is 30 bricks, where we consider all to be sugars. This concentration will be equal to 182 kg per meter cubic for our enzyme concentration. They also determine the optimal to be 6 kg per meter cubic on a dry basis. The mass needed for the input is presented. This was calculated using the volume calculated, which will be discussed in the next section.
Considering that the concentration for our substrate is based on the pure component, we have that our raw material is a solution. We will need to determine the total mass of molasses using the mass relation. Here we have that the 30 kilograms of sugar are the 57.2%, and we want to obtain the 100%. Then we obtain that the 100% is 52.45 kilograms of molasses. For each independent reactor, we have that the mass of the sugar is 2,412.0915 kilograms of sugar. And for the mass of the molasses, we have that is 4,217.14 kilograms. Then we multiply the mass calculated by 8 because the previous calculations only considered the production for a single reactor, but we will have a system of 8 reactors working independently from each other. So we have a total input from the mass is 33,737.12 kilograms, and from the enzyme is 2,120.52 kilograms. For this raw material input, we consider that the molasses is first added to the water, as all the sugars are the only soluble solids, and then it's filtrated, remaining only the sugar solution with 30 bricks. Output. For the mass of the product, the remaining substrate, and the total growth of the enzymes, we repeat the same process multiplying for the volume. So for each independent reactor, we have that the mass of the enzymes is 277.876475 kilograms. The mass of the sugar is 2,636.071425 kilograms, and the mass of the ethanol is 2,673.6223 kilograms. The results obtained will be multiplied by 8, but in addition we calculate the mass produced of the CO2 by using the mass stoichiometry relation between ethanol and CO2. And as a result, we obtain that the mass produced of CO2 is 20,432.57954 kilograms. To calculate yield from sugar to alcohol, we use the next equations. The first one is the yield from the total sugar present in the mixture, and the second one is from the total sugar consumed. In the first equation is the final ethanol concentration minus the initial ethanol concentration times 100 divided by the initial sugar concentration times 0.511. This value is the maximum theoretical conversion at 100%. For the second equation is the final ethanol concentration minus the initial ethanol concentration times 100 divided by the initial sugar concentration minus the final sugar concentration times 0.511. Using these equations, now we know that a total sugar present in the mixture is 65.0738%, and the sugar consumed is 96.8155%.
First of all, está bonita, güey. First of all, we need to define the design equations for the reactive system. We know that for a batch reactor, there will be no flow, there will be no input flow or output flow. There will be only accumulation depending on the formation of the products and the separation of the reactives. We have that for a bioreactor, the equations that define the accumulation will be depending on the cells, the substrate that can be considered the food for the cells, and the product, that is the product formation depending on the substrate. In this case we have sugar that will be converted into ethanol. In addition, we have that the parameters needed to solve the equations will be this right here. Cp is considered the maximum concentrations of product at which the cells stop producing more product. The other constants are depending on the system. The Ks is considered the monod constant. Mu maximum is considered a constant that will be depending on what is the maximum velocity at the formation of the cells. And we do know that these equations take place at the same time. So the system will be a differential equation system that need to be solved using these parameters. First, we need to define what are the constant parameters. Not all the parameters that we are using are depending on the concentration. Some of them are, some of them not. No. So we have here these parameters that are constant. These are defined by the system, mostly on the cells or the enzyme. In this case, we are using the same enzyme that is utilized in the production of beer. And from experimental data obtained by Siqueira et al., we obtain some of the other parameters that are needed to solve the reactive system, which will be in the lower part. We can see them. Then, from what we have learned in class, we know that there will be certain parameters that are depending on the kinetics of the reaction. These parameters are the mu maximum and the Ks, which is the monod constant. So to solve them, we need to use the experimental data shown before, and we have to apply a nonlinear regression utilizing the Hannes-Wolff form for the monod equation. We first apply a nonlinear model using the software Polymath to obtain an initial estimation of these values. The initial guess values we use are obtained from Microsoft Excel. So we put the initial guess and we obtain some values. Then we fix the mu maximum in the equation because we know that it has a lower value in the 95% confidence test. So it will be okay to fix this value and continue with the calculations. Then, now that we have fixed our mu maximum value, we need to obtain the Ks from the second nonlinear. We apply now a second nonlinear regression utilizing this fixed value in the form of the Polymath model. We can see that is fixed right down here. Then we obtain a Ks value with a lower percentage of confidence in the test. At last, we make an analysis to know if our calculations are correct. From what we can see, we have an R square of 0.94 and an R square adjusted at the same value. We know that the R square adjusted is more confident, has a more confidence value than the R square in simple form, but they are the same, so we can use both. And we know that for an R square greater than 0.9, our value will be correct. However, it has not it is not very approximate to 1. So there will be certain parameters that we are not taking in account in the resolution of our system. But it is difficult to obtain these parameters, which are more about the behavior of the cells. For what we know, we are chemical engineers, so we do not wish to know all these kind of parameters. So it is correct for us to assume these constants for this model in the simple form are okay. Then we produce a graphic for our final concentrations, and we can observe in the blue graphic right down here, this is the cell's concentration over time. We know that the cells will be producing more ethanol during the exponential growth phase, which is what we can see here. Right at 11 hours, the exponential growth starts to decrease, so it is good to stop the reaction at this point, and we will have a high enough formation and a high consumption of the sugars we are feeding to the system.
After the system is solved, we obtain the final concentrations. Using the equation of the productivity for a bioreactor and the desired production, multiplying the time needed to carry a single reaction, we obtain the total volume. We determine that the best quantity of reactors to distribute this final volume is 8 batch reactors. So we divide the total volume by 8, and we consider the specification for a bioreactor not to be filled at more than 80% of its capacity, so the final volume will be 60 cubic meters with an altitude of 3.5 meters and 4.67 meter diameter. In conclusion, as a team, we think that these calculations do make sense because we have seek for experiences where we can observe different processes for ourselves. In some of them, we have had business visits to a variety of ethanol production plants, and based on the observations during our visits, the team affirms that the calculations made are correct, because most of these plants have 4 to 8 batch reactors working simultaneously to achieve the desired quantity of the product, which is very critical for the alcohol industries. At the same time, these reactors have height of 2 average human adults, and it takes the size of a whole classroom to feed them. Also, we don't know our calculations are correct because in order to produce a large scale, even considering a high rate of conversion, you do need an immense reactor for these productions.